So now that we've talked a little bit about membranes and what's surrounding the outside of the cell, I want to dive in a little deeper on the inside of eukaryotic cells and some of the specialized organelles and other structures that we see inside them. So first, I've mentioned the cytoplasm a couple of times as we've been talking about cells, but just to make sure we're all on the same page, the cytoplasm is everything inside the plasma membrane. So we have our cell, our extracellular environment. So the outside, we have our cell membrane. And then inside that membrane, we have cytoplasm, and it's mostly made up of water. Like 70 to 80% of it is water, but it also has a bunch of different proteins floating around. So it's semi-solid if you were to be able to touch it. Now, as we start to talk about some of these organelles in a eukaryote, I want to think a little bit about how the proteins that we've seen embedded in membranes are related to some of these structures, because several of our organelles are actually going to be pretty specialized for making and modifying proteins, which is why we've waited until now to talk about them. So our nucleus is one of those structures. So hopefully it's familiar to us from the first day of class. We remember that a nucleus is specific to eukaryotic cells. So we don't have one in prokaryotes, but eukaryotes have a membrane-bound nucleus, which means we have our DNA clustered inside, and it's surrounded by a membrane. So now that we know that membranes are really good at controlling what can cross them, they're selectively permeable, so they can control what goes in and what goes out, we can kind of understand why that would be important for a cell. So this membrane around the nucleus keeps the DNA in because DNA is too big to get through that membrane. And it also keeps random ions and other molecules from coming in and messing with the DNA. So it also protects the DNA, keeps other stuff out. Okay, so here we can see on our structure, we have a nuclear membrane. Here we see the DNA. We have another structure called the nucleolus, which is related to production of ribosomes and RNA. We can also see that our nuclear membrane here has some little pores. So that membrane controls what goes in and out. We'll see in our next unit that it's actually really important to get information about that DNA out into the rest of the cell. So we don't let the DNA leave, but we do want to let messages about the DNA get out into the cell. And that's because the information encoded on that DNA on different genes, which are basically little chunks of DNA, we have information about proteins that we want to make. So like those transport proteins embedded in the membrane, you're going to have a gene in the DNA that codes for each of those proteins. So if we were to start making a protein, we'd start by reading a little chunk of that DNA. Next to the nucleus, so here we see our nucleus, here our DNA is pictured in red, we see the endoplasmic reticulum. So the endoplasmic reticulum is also made of membranes. So these membranes happen to form a sort of network of tubules, and we have a portion of the endoplasmic reticulum that is rough because it's studded with ribosomes. So ribosomes, we're going to learn, are very important for making proteins. So our rough endoplasmic reticulum is studded with ribosomes. And then we have our smooth endoplasmic reticulum, which has no ribosomes. So these are the two parts of our endoplasmic reticulum. Now, if we're talking about making proteins, the part we would focus on is this rough endoplasmic reticulum. Because as we're making a protein, making proteins actually happens at the ribosomes. The ribosomes are really what makes the protein. So part of the job of this rough endoplasmic reticulum is to help make proteins. So they're involved in protein synthesis and sorting, and are where protein folding happens. So we talked about the different levels of structure of a protein. So one of the places that they're going to be folding up and taking shape is in the endoplasmic reticulum. The endoplasmic reticulum is also important because it makes those phospholipids that make up the membrane. So remember that our membrane is a phospholipid bilayer. So we have those two levels of phospholipids with their fatty acid tails facing each other. Now those phospholipids are being made in the rough endoplasmic reticulum. The smooth endoplasmic reticulum doesn't do those same things. It doesn't have ribosomes, so it's not involved with proteins. It also doesn't make phospholipids. Instead, we're going to see carbohydrates, other lipids, specifically like steroid hormones being made by the smooth endoplasmic reticulum. They're also important for storing some important ions that the cell needs for various processes like calcium. So the rough endoplasmic reticulum is making stuff like proteins, phospholipids. The smooth endoplasmic reticulum is making some carbs, making some steroids, and storing stuff. As we move away from the nucleus, so right next to our nucleus, we had our endoplasmic reticulum. Next, we're going to see a structure called the Golgi body. So the Golgi body, again, is made up of membranes. And these membranes are going to look kind of like a stack of pancakes. So our ER looks kind of like coral, basically. Our Golgi looks like pancakes. Golgi apparatus, Golgi body, Golgi complex, or just Golgi all mean exactly the same thing. So anytime you see one of those terms, talking about this structure. Okay. So this structure is important because it modifies proteins that come through it. So you can see here that the rough ER, the rough endoplasmic reticulum, is in this drawing. You can see we have these little sacs coming and merging into the Golgi. So we saw when we were talking about endo and exocytosis that we can have little bubbles of membrane hit other pieces of membrane and then fuse into them. And we can also have little bubbles of membrane bud off of a membrane. So that's what's happening here between the rough endoplasmic reticulum and the Golgi. So we had proteins here partially being made by these ribosomes and the endoplasmic reticulum, getting folded perhaps. And then they're going to leave the endoplasmic reticulum by having these membranes bud off, creating structures called vesicles. So anytime we see a little bag of membrane with stuff inside it, we're going to call that a vesicle. And then these vesicles travel over to the Golgi, merge into it, and the Golgi is going to continue to modify those proteins. It's going to sort them, so try and figure out where they have to go next. And then it's going to package them into more vesicles and send them on their way. So as we were talking about exocytosis, we talked about vesicles merging into the plasma membrane. So we can see here in this example that this is what the Golgi happens to be doing with this sort of black protein here. It's creating a vesicle and sending it off to the plasma membrane to release outside the cell. So we might refer to these processes as protein sorting, that's figuring out where they're going, processing, it'll be tagging them with a little extra sort of, kind of like postage stamps telling the protein where it's going, and secretion. Secretion just means that it's sending stuff to the outside of the cell sometimes, as well as to other locations within the cell. So here's a map of basically where proteins are made and how they get sorted. So we're going to ignore this part here on the left. This part here on the left is just stuff that gets made entirely in the cytoplasm in what we call free-floating ribosomes. And then those proteins just go directly to their targets. But here we can see the path that we've just talked about that some of our proteins take. So in this process called co-translational transport, we have proteins that start to be made on ribosomes in the cytosol based on information in the DNA. And then they get shuttled to the endoplasmic reticulum, which has more ribosomes. So they continue to be made at the endoplasmic reticulum. They get packaged into little vesicles and sent to our pancake structure Golgi. And then the Golgi modifies them does some processing steps, and sends them off to where they're going, which may be to the outside of the cell. So we may be sending them into the extracellular environment through secretion, which is a form of exocytosis. Or we could be making proteins that are perhaps transport proteins, part of the plasma membrane itself. So the Golgi could send them to the plasma membrane to get embedded in it. We could send it to other parts of the endomembrane system. So we could basically send these proteins anywhere to anything with a membrane. Or we could leave these proteins in a bag, at which point we'd call them a lysosome, which is another specialized organelle. It's specialized for digestion. So let's take a look at that lysosome. So the lysosome is basically like a specialized vesicle. So it's a membrane, so a little bag with some proteins in it. And specifically, those proteins are going to be digestive enzymes. So the lysosome acts like the stomach of the cell. So it does two different kinds of eating or digesting, basically. It does phagocytosis, which is eating food, so anything that the cell has maybe taken in and wants to break down for energy. And it can also break down worn out organelles. So it's kind of like if we want to reuse the parts from one of our other organelles, we don't want to waste our phospholipids or something. A lysosome can break down those organelles so that we can reuse those molecules for something else. And we want to contrast our lysosome with a structure called a peroxisome, which also breaks stuff down, but it's not really breaking stuff down for potential reuse, but it's going to be breaking down molecules that are toxic to the cell. It's specifically going to be creating hydrogen peroxide as a byproduct, which is just to say it's going to have some like intense chemicals that would be harmful to the cell that it's producing as it's breaking down toxins. So it's really important that this is also all surrounded by a membrane. So peroxisome also looks a little like a vesicle, kind of like a little baggie with some stuff inside. That brings us to our semi-autonomous organelles. So these aren't involved in creating proteins or really using those proteins, but they're mostly going to be involved with energy. We call them semi-autonomous because they're a little special. They kind of do their own thing. So mitochondria in animals and chloroplasts in plants each have their own DNA. Mitochondria even have some of their own ribosomes. They can grow and divide, make more of themselves on their own without the rest of the cell, which makes them partially autonomous, which just means that they're under their own control. But they're not fully autonomous because they still depend on the cell to make other parts, like their membranes. So they have a sort of interdependent relationship with the other structures in the cell. So let's look at a mitochondrion a little more closely. So mitochondrion is one, mitochondria is all of them. If I go back and forth between those words, that's just singular and plural. So the mitochondria, their primary role is to make ATP, which we've seen is the type of energy our cell uses. So we saw that in primary active transport, we need ATP to fuel the movement of molecules against their gradient. The mitochondria are where we would get that ATP from. So the mitochondria are made of two membranes, basically. So they look kind of like a bean. There's an outer membrane and an inner membrane. There's a space between those two membranes called the intermembrane space. There's also a space inside that innermost membrane, which we call the mitochondrial matrix. Now, all of these spaces and both of these membranes are going to be important when we start to talk about metabolism at the end of this course, because we're going to hold different molecules and different processes inside the mitochondrial matrix versus in this intermembrane space. And we're partially making ATP by controlling the production of gradients and then their release. So we're not going to talk about that in detail right now, but it's really important that the mitochondrion has these two spaces that are separated from each other by membranes, because that means that we're going to be able to build up different concentrations of molecules, which is going to be really important for our process in metabolism when we start to talk about that. We're not going to talk about chloroplasts on their own. They are present in plant cells, and we'll take a look at a plant cell on the last slide. The theory of endosymbiosis, though, refers to both mitochondria and chloroplasts, and it's really a theory for why your cells have these. Because these are kind of weird organelles. The fact that they have their own DNA is super weird. So the hypothesis for why we have mitochondria and why plants have chloroplasts is that maybe way back in distant past time, mitochondria and chloroplasts could have been their own cells, their own little prokaryotic cells that were specialized for energy production, basically, that were really good at making energy, and that they got absorbed by a bigger cell. Maybe that bigger cell wanted to eat the mitochondria or chloroplasts, right? But it realized that they were really useful. So instead of breaking them down using a lysosome or something, the mitochondria and the larger cell developed this symbiotic relationship where the larger cell makes different parts for the mitochondria, and the mitochondria makes energy for the larger cell. That's the idea of what could have happened to get us to this system we have today, where the mitochondria are not their own cell. They're just an organelle, but they're weird because they have some components that you would only really expect to see in an independent cell. So our possible evidence for this theory is the presence of their own DNA that's different than DNA in the nucleus, the fact that they have their own ribosomes. They're actually about the same size as prokaryotes. So like they're about the same size as a bacteria. The cytoskeleton is a structure in the cell that we don't usually draw in, but is really important to how cells function. So the cytoskeleton is basically just a network of proteins. Its job is to maintain the shape of a cell, to hold the organelles in place. And as it's holding stuff in place, when it shifts shape slightly, that lets things move. So it lets organelles and things within the cell move, and it also allows cells to move. So we can see here the edge of a mitochondrion. Here we see the edge of the endoplasmic reticulum. We can tell that because it's the rough ER, so we can see these ribosomes. Here we see a vesicle. And here we actually have drawn on the cytoskeleton. So we can see that it's forming like this scaffolding, right? So like when they're repairing a building, you create a scaffolding so that you can work on it. So the cytoskeleton is kind of like that, but it's there all the time, holding things in the cell in place. We have a couple of different parts of the cytoskeleton with different jobs and different sizes. So you can see that we have these yellow strands, these pink strands, and then these sort of light red strands. Those are all different parts of the cytoskeleton. They're all important. So basically all of them do a little bit to help cell shape and support. So hold the cell in its usual shape. Different cells are different shapes. We've always been drawing this circle one, but sometimes you'll see some boxy ones. Sometimes you'll see some sort of teardrop shaped ones. And that shape will be dependent on what the cytoskeleton does. But two of our types of protein filaments are going to have very specialized jobs as well. So our microtubules are going to be important because they're going to help us move things around within the cell. And we'll see that that's going to be specifically important when we get to our genetics unit and start to talk about cell division. The microtubules are really what's going to allow our cell to take different pieces of DNA and pull them apart as they start to create two new cells. Our intermediate filaments, we're not going to talk about any of the specialized jobs they do in this class, so we're just really going to think about them as being really important for cell shape and support. Microfilaments are our last type of protein filaments in the cytoskeleton, and their special job is really for cell movement. So you'll talk about them next year when you start to talk about muscles, for example. Your microfilaments will be really important. Okay. Looking at that first type, I said that microtubules would be important for cell division. So a structure that happens to be made of microtubules is our centrosome. So our centrosome we can think of as an organelle that's basically going to make like a net or a rake that lets us pull apart chromosomes when a cell is dividing. It's made up of a bunch of microtubules grouped together. So each of these churro-like things is something called a centriole, and two of them together at 90-degree angles makes a centrosome. So we'll see those again when we talk about cell division. Some other structures made of microtubules are flagella. So like on sperm cells, there's a whip-like tail. That's made of a bunch of microtubules bound together. And some other cells have something called cilia, which are similar but shorter. There are many of them. So they can gently move as well to allow cells to move in their environment. So all of these would be made of microtubules. So that's a little sort of deeper dive on some of the specialized functions of these protein filaments that are also part of our cytoskeleton. Finally, we're going to look at a couple different things that we see in plant cells. So these you won't really be expected to know for this course, but you should, in general, after having taken a cell biology course, have heard of a couple things about plant cells. So one important thing that a plant cell has that an animal cell doesn't is a cell wall. So surrounding our membrane, a plant cell still has a plasma membrane. We have a hard, rigid cell wall. And we talked a little bit about that when we talked about osmosis. So it can cave in a little when the cell is in a hypertonic environment. But when we're in a hypotonic environment, it gets very rigid. And that's actually because of the action of this next structure we see in plant cells, but not animal cells, our vacuole, which is like a reservoir of water inside the cell. So when it's big and full, everything gets pressed out against the cell wall, holding it in a nice rigid shape. When it gets really small, the cell wall starts to cave in. And we have our chloroplast, which is another type of semi-autonomous organelle. So your plant cells do have mitochondria, but they have an additional type involved in photosynthesis. And then your plant cells also have something called plastids. So you've seen that plants are all kinds of crazy colors. That's partially because of our plastids that